Hello all. Today we are going to move forward with some very important concepts. And today we are going to take a look of the various terms related to Operon. First of all, what is an Operon? An Operon is a coordinated system of genes in which all the genes involved in a particular function or all the genes involved in a particular metabolic pathway, they are present exactly adjacent to each other in such a way so that where one gene ends, immediately starts the next one. Example, in lac operon, here lac means lactose, okay? Operon, here this is a combination of three important genes, Z, Y and A. The Z gene is responsible for beta-galactosidase, Y gene for galactoside permease and A gene for thioglycoside transacetylase. Now, all these three genes are extremely essential for E. coli if E. coli want to use lactose as a source of energy. Okay, and that's why these three genes should be expressed. But you can see here these three genes are present exactly adjacent to each other. There is not a gap. This is not the case in eukaryotes. In eukaryotes, all the genes involved in a particular function might be present in all different chromosomes. So it's not there that immediately after where one gene ends, immediately starts the next one. But in prokaryotes we have this system. And such coordinated system of genes which can be regulated, which can be expressed at a time, and this is called the operon. Here you can see this is the promoter, P for promoter. This is the operator. Here there are, in case of lac operon, we have three operators, O1, O2 and O3. Okay, so this is the promoter. And on promoter will bind RNA polymerase, correct? So suppose on this promoter is present RNA polymerase. This is the RNA polymerase enzyme. Of course, what the RNA polymerase is going to do? The RNA polymerase is going to express these genes. Here we have written +1. +1 means the first nucleotide which will be expressed, and from these three genes shown in lac operon, there might be many genes, much more than these three genes, okay? But these all the three genes, they will be expressed in the form of a single mRNA. And this mRNA, it will have the sequence of Z, that is beta-galactosidase, Y, as well as A. And that's why this mRNA is called polycistronic or polygenic mRNA. Okay, so this mRNA which is formed will be polycistronic or polygenic mRNA. Okay, this is not the case in eukaryotes, as eukaryotes don't have operon system. A single gene will be present, and from a single gene will be formed mRNA which will be only monocistronic or monogenic. Correct? Now, in lac operon, you can see many factors are there. Okay, if we will come on this side of the same operon, you can see there is a I gene and there is a promoter of I gene, and of course if there is a promoter will bind what? RNA polymerase, and this RNA polymerase will show transcription of this I gene. Okay, now this I gene after transcription will form its mRNA which on translation will form a repressor. Repressor, a protein which won't allow the gene to express. It will repress the operon. Okay, and that's why the repressor is formed. But you can see the repressor is an active repressor. Now, what is an active repressor? A repressor which won't require anyone else to bind on operator. So, the repressor immediately after its formation will go and bind on this O1, that is the first operator, because this operator is having higher affinity for repressor. So definitely it will go and bind here. Now, until and unless your repressor is bound on the operator, your RNA polymerase can't move forward for expression of this Z, Y and A genes, means lac operon cannot be expressed. So suppose this is your promoter region where RNA polymerase might have been bound, and here there is operator, correct? And on the operator is bound your repressor. So, and from here will start the functional genes. So definitely until and unless this repressor is bound on the operator region, your RNA polymerase cannot move forward. So this is what a repressor is. What a repressor is? Repressor, a protein that has negative effect on gene expression. Means it won't allow the RNA polymerase to go for transcription. Now, repressors can be DNA or RNA binding. Here you can see this repressor is a DNA binding repressor and is not allowing transcription of lac operon because your RNA polymerase cannot slide forward as its path is blocked by repressor. Correct? In case of RNA binding repressors, what they will do? Definitely they will block the path of ribosomes, because until and unless your ribosome will bind on your mRNA, they can start translation. So its path of these ribosomes is blocked by those repressors which are RNA binding repressors, because their aim is not to express the gene, okay, or operon. So here this repressor has done that, and this is an active repressor. But another site of the same repressor, in case of lac operon, this site of the repressor is having affinity for the second operator, that is O2. Now you can see O2 is present amongst the Z gene, the gene responsible for beta galactosidase. The O2 is present amongst that. So O2, that is operator 2, is a part of the Z gene, as well as it is having affinity for this second site of the repressor, and that's why this O2 get attracted towards this repressor, and that's why this complete operon will fold like this. You can see like this. If it will fold something like this, you can see here the repressor is bound on O1. This is the O1 and the repressor is bound. And this O2, this is Z gene, and O2, this part, okay, this part is now bound on O2. And thus forming a loop, or I can call it a twist. And because this twist is formed, you can see the promoter region is now hidden behind this region of the DNA. Okay? Because it is the loop which is formed. And that's why the promoter region, you cannot see it. It is present behind this part. And if RNA polymerase won't be able to see promoter, it won't be able to bind on promoter. Correct? This is how the gene is repressed. Okay? Means this repressor is very effective type of repressor, because it's an active repressor. It don't need anyone else, but other than that, it's forming a knot-like structure. Correct? Repressing the gene completely. This is what the repressor is going to do. And this type of repression is actually called negative regulation. So what is the negative regulation? Everywhere where the gene or operon is repressed. Correct? But lactose, when it is present in the medium, because lactose here is the inducer of lac operon, definitely E. coli will express the genes of lac operon when lactose is present in the medium. So suppose now lactose is present in the medium, what the lactose will do, this part of the repressor, this one, is having affinity for lactose. So whenever lactose is present in the medium, this lactose will go and bind on this repressor and remove it. So the repressor which was active is now converted into inactive repressor. Of course, if the repressor will come out because of some conformational changes, it will lose its ability to bind on operator, and that's why it will come out, and definitely this complete operon will get straightened. And now this, as it is straight, it is open, definitely who will bind on the promoter is RNA polymerase. And repressor is removed, and thus the RNA polymerase will start its gene expression from +1, and a polycistronic m-RNA with Z, Y and A will be formed. So what lactose is? Lactose will be an inducer. Inducer, somebody who will induce the gene expression. Correct? So here inducer. What is the inducer? Something which will induce gene expression. Generally, it converts the active repressor into inactive in case of prokaryotes. So in case of prokaryotes, just now we have seen the active repressor is converted into inactive by lactose in case of lack of operon. But in case of eukaryotes, in eukaryotes, operon system is not present. Okay? But in case of eukaryotes, if we will talk about inducer, somebody who can convert the inactive activator into active. There are some activators in eukaryotes. And if the activator is inactive, it can be converted by inducer into active repressor. Okay? But that is only in case of eukaryotes. So, what lactose has done? Lactose has induced gene expression. Correct? This is what is called positive regulation. Whenever gene is expressed or operon is expressed, that type of regulation will be referred as positive regulation, and what lactose has done here is a positive regulation. Correct? Now, other than this, keep in mind, simply presence of lactose is not sufficient for strong induction of the lac operon. Why? Because if glucose is present along with lactose, definitely E. coli or any prokaryote will always try to utilize glucose as a source of energy, and even if lactose is present, it won't express the lac operon. The reason is very simple. It don't want to use lactose. But consider if the glucose concentration will go down, so the glucose sources get depleted. In such case, the E. coli or any cell will try to use now lactose. So lactose is present in the medium, and the lactose might have removed your repressor by converting the active repressor into inactive, and your RNA polymerase will have a chance to bind on the promoter. But this promoter of lac operon is actually a weak promoter. Weak promoter means though promoter is present, it might not have high affinity for RNA polymerase, and that's why it's not essential that immediately the RNA polymerase will come and immediately the gene expression will take place. For that, what do we require is the help of some other auxiliary protein. When we talk about auxiliary protein, means we are talking about secondary proteins. Okay? What auxiliary proteins are? Auxiliary proteins helps RNA polymerase to recruit on promoter. Because in case of lac operon, the promoter is weak. So even if we don't have repressor, the RNA polymerase might not immediately come. But as the glucose concentration will go down, cell will start forming cyclic AMP. As the glucose concentration will go down, cyclic AMP concentration will increase, and there is a protein called cyclic AMP receptor protein, CRP. And this, as the name suggests, it's a cyclic AMP receptor protein. So on it will bind this cyclic AMP which is just now formed. And this cyclic AMP CRP complex has a site at -40 and -60 region. Minus means the region which is present upstream of the functional genes. Okay? So if this is the +1, the first nucleotide in front of it will be -1, -2, -3. In this way, at the -40 and -60 regions, you have two CRP binding site. CRP is the cyclic AMP receptor protein. But this CRP itself cannot bind until and unless cyclic AMP will bind on it. Now, this cyclic AMP CRP complex can go and bind on these two sites. When this cyclic AMP CRP complex, such two complexes will bind here. they will increase the affinity of RNA polymerase for promoter. So, on the promoter, RNA polymerase will bind, but its affinity for promoter will be increased by binding of these two, cooperative binding of these two CRP, that is cyclic empty CRP complexes. And that's why now binding of this will increase the affinity and the RNA polymerase will get recruited on promoter. So this is what auxiliary proteins are going to do. They are secondary, but again it's a part of positive regulation. The reason they are helping the genes to express. Okay, so this is what is auxiliary protein, which will help the RNA polymerase to recruit on promoter, and this is also part of positive regulation. So strong induction of lac operon requires presence of lactose and absence of glucose or quite low quantity of glucose in the surrounding of the cell. Correct? Now the gene expression will take place at a faster rate. Correct? So this is what is positive regulation has been done. Initially it was negative regulation. But presence of lactose and absence of glucose, which will cause cyclic empty CRP complex to form, this binding, okay, and presence of lactose will strongly induce the gene expression. And here lactose will be the inducer, but cyclic empty CRP will act as auxiliary proteins. Now, sometimes the repressors are inactive, just like in case of tryptophan operon. In tryptophan operon, what do we have is a promoter, an operator, and here there is a +1, and after the +1 there are EDCBA, such genes which will give rise to certain proteins which will help in forming tryptophan as as it is a very important amino acid, because E. coli has genes to synthesize all the 20 amino acids. In a very similar way, this is a tryptophan operon or TRP operon, and it has genes. If these genes are expressed, if these genes will be expressed, here there are functional genes. If it would have been expressed, they will lead to formation of tryptophan. But in the same operon, you have R gene, and this is the promoter of R gene, that is represented by PR. On the PR will bind again. Of course, it's a promoter, so who will bind? Definitely, it will be the RNA polymerase who will bind it, and expression of this R gene will take place. Its transcription as well as translation, and what is formed is from R repressors form. But this repressor is inactive repressor. In lab, you have active repressor. This repressor won't require anyone. It will directly go and bind on operator. But this repressor is inactive, and being inactive, it won't bind by itself on the operator. But if tryptophan is present in the medium, definitely if you have tryptophan, you don't need to synthesize tryptophan. Then two such tryptophan molecules should bind on the inactive repressor, converting the inactive repressor into active. And this active repressor will now go and bind on the operator and won't allow the genes to express. What has happened? This is what? This is a negative regulation. But your repressor here is inactive. And what tryptophan has done? Tryptophan has bound to the repressor, converting the inactive repressor into active. So tryptophan has also given a negative regulation. It had not allowed the gene to express or the operon to express. Such molecules are called co-repressors. So here tryptophan is a repressor means which is not allowing the genes to express. Co-repressor, which is not directly binding on the DNA, but binding the inactive repressor and converting it into active, and thus won't allow the gene to or operon to express. Look at it, co-repressor. Small molecules in case of prokaryotes. These are small molecules like tryptophan, or proteins in case of eukaryotes. Again, in eukaryotes, operon system is not present, but co-repressor in case of eukaryotes are generally proteins most of the time, which convert inactive repressor into active repressor and thus won't allow the gene or operon to express, and generally they don't bind to DNA directly, they bind to inactive repressor, but they are helping in repressing the gene. They are not allowing the gene to express. This is a type of what? This is also a type of negative regulation. Okay? So these are the various terminologies regarding the operon system. Okay? We have seen about repressor. Okay? Somebody who won't allow the gene to express. Co-repressor, which will also show negative regulation, but not by binding to DNA. It will bind to the inactive repressor and convert it into active. We have seen about inducer. Lactose is the inducer of lac operon because it has converted the active repressor into inactive. Correct? So definitely allow the gene to express. But in case of weak promoters, RNA polymerase will require much more time to bind on the promoter. In such case, you might require auxiliary proteins like the cyclic CRP complex. Auxiliary, which will help the RNA polymerase to recruit on promoter. And then we have seen about inducer, okay, as well as positive and negative regulations. So this is all about the various terms regarding operon. Thank you.